This is an introductory conference, so for those of you who are looking for the cutting edge of the cutting edge, you have to come to our weekly seminars instead. I think that the conference we have next year will do a lot more of the cutting edge of the cutting edge. But at this meeting, we're going to talk about actually implementing some of the basic findings over the last 10 or 15 years into clinical practice. And Brian Decker is going to talk about some of those to begin with. And then I'm going to talk a little bit about how we, and you and everyone, should go about choosing the tests and implementing the tests that we use. And then later on in the day, we have some of our academic superstars here, people like Jamie Remberger, Brian Schneider, Samir Gupta, clinician scientists who are going to talk about the work there in their specific areas. But with no further ado, I'll invite Brian Decker to come up and start off. Brian's assistant professor in the Division of Nephrology and Clinical Pharmacology here at IU, and he's going to begin.
Thank you, Dave. All right. Well, thank you all for coming today. Let me see if I can get this figured out here. All right. As the title suggests, I'm going to give an introduction to clinical pharmacogenomics. And please, at any point during this talk, if something doesn't seem right or you're confused, just stop me. Otherwise, I will just keep talking and talking. What I hope to talk about today is a case. This will hopefully get us all in the mood for this and get us oriented. I'm going to talk about this fundamental change in the medical paradigm, and I think paradigm shifts have probably been overused and kind of a cliche, but I think clinical pharmacogenomics and what it's permitting us to do now with patients' medicine, this certainly applies. Short primer, and this is a short primer, just a few slides, but really got to be able to speak the language in clinical pharmacogenomics to know how to approach your patients with this. Talk about SNPs, which are single nucleotide polymorphisms. You'll be hearing this word a lot today. Go back to clinical pharmacology a little bit and talk about metabolism and transport, because this is what really impacts clinical pharmacogenomics, and the SNP nomenclature, which, like every nomenclature system in medicine, is perfectly easy and logical, right? Well, it's got its issues, but we will wade through that. And then I'm going to talk about some pharmacogenomically important medications, and I'll stress again and again, this is just a small grouping. Some of these already made it into clinical translation as far as clinical pharmacogenomics is concerned. Others are still in the research mode, but it's also a good sneak peek at what we're going to be talking about in much more detail later today. And then we'll get back to the case and see what we learned this morning, all right? So here's our case. RF is a 58-year-old female, past medical history of coronary artery disease, hypertension, hypercholesterolemia. She comes to see her heart doctor with shortness of breath and a sensation of a racing heart. She has atrial fibrillation. Part of her treatment plan is oral anticoagulation initiation with warfarin. All right, RF has been reading the internet. She knows all about clinical pharmacogenomics. She actually has enough foresight to agree to a genetic screening for her because she knows this may be able to affect her warfarin therapy. And this is what she gets back. She's heterozygous for CYP2C9 star 2 star 3, and she has this GAV core C1 genotype. And I promise you at the end, all this will make sense, okay? And we'll know how to handle this patient. All right, let's talk about the current medical paradigm. And this is an oversimplification. This is maybe the Decker view of the world, but I think it gets the point across here. And medicine is all about disease. And this is what we're in the business for. We want to mitigate and cure disease, correct? And to do that, we need research, and we need lots of money for research. But the end result is hopefully this golden magic pill, this capsule that'll take care of the disease. So we give this to our patients, and we cross our fingers and hope for an outcome, all right? And of course, this is my salute to Big Pharma, and I need to draw another arrow because that's where a lot of this money comes from. But our patient outcomes, mitigate or cure disease, this is obviously what we're shooting for. This is what we want to always happen with our patients. But sometimes it doesn't. Sometimes we don't have any effect. Sometimes we give a patient our favorite medication, and then we're astounded when it doesn't work. Worst case scenarios, patients have an adverse drug event. And these are sometimes very difficult to predict. Many, as you know, are idiosyncratic. We don't know what kind of reaction a patient's going to have, and sometimes they're serious, unfortunately, if not fatal. The problem with the current medical paradigm is that we take these magic wonder capsules, we treat all of our patients if they're genetically identical to each other, that every patient has the same risk for good efficacy or chance for efficacy, and every patient has the same risk for an adverse drug event. We tend to treat our patients as if they're all clustered about this mean here. Okay, this is the mean of good effect and low adverse drug events, when we have many, many patients who obviously lie out here. Now, the pharmacogenomic medical paradigm, and this is where the shift comes in, is that we don't treat patients as genetic clones of each other. We treat every patient as if they're genetically different from each other, because in reality, we all are. And we do a genetic test, and we try to match their medication with their genetic profile here, so that our patient outcomes are more mitigation and cure of the disease, and ideally no serious adverse drug events. We don't always hit this mark, even with pharmacogenomics, but that's certainly our goal. And this is the whole concept, you'll hear this again and again, of personalized medicine. To put it another way, we know within this large group of genetically diverse individuals, we've got a group who we know we can cure or at least help, and we have a group that, and hopefully always a smaller group, that are more likely to have an adverse drug event. So we do a genetic test. We get their profiles, we get their pharmacogenotypes, and this allows us to identify the phenotypes, allows us to predict the phenotypes. So this group we're going to give the drug to because we know that they're going to have a good response. This group we're going to find something else because we don't want them to have an adverse drug event, because again, our first precept is to first do no harm, as you all remember. Clinical pharmacogenomics is not new, actually. Certainly most of the progress has been made in the past 10 years, 15 years perhaps, but it's not a new concept. We go back all the way to World War II. African-American soldiers given primaquine had hemolysis, and this led to the identification of the G6PD deficiency. Later, in '57, Kalo and Gunn found subjects homozygous for a gene encoding the atypical form of pseudocholinesterase, which had the delayed recovery from succinylcholine. And we're still using this test today. And there is Dr. Kalo right there. Interesting guy. I don't have time to talk about him, but very, 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 very cool history. In 1959, Vogel and others looked at a genetic variation in N-acetyltransferase, and they found marked differences in the drug half-life of isoniazid and the plasma concentration of isoniazid. And this was associated with hepatotoxicity. Eichelbaum, 77, identified the CYP2D6 polymorphism. Now we have more than 100 alleles for this. 2001, this is a very key study. Drucker demonstrated remarkable response using imatinib for CML. This is in cancer chemotherapy. In 2003, the finished version of the human genome sequence is completed, obviously very big, and this past summer. I think this is also a big milestone. The lawyers are probably still sorting out what the ramifications of this is, but the U.S. Supreme Court, as you probably know, ruled that human genes cannot be patented. I think this will largely be a good thing for clinical pharmacogenomics. Maybe not so much if you're a biotech company right now, but certainly good for us. And then what I like to call a moving milestone is the cost of human genome sequencing. This is just amazing. It took 13 years and $3 billion to get the sequence first done in 2003, and now we can do it in less than 2 weeks and at about $1,000. And this curve is probably even lower now, but it's amazing. It's even outpaced computer technology advances, which is this whole Moore's law. So this is definitely driving the bus as well. So let's switch gears a little bit and talk about clinical pharmacogenomics. We're certainly more alike than we are different. 99.5%, which is just amazing to believe, of a genome between any two individuals is identical. And as you know, mutations that occur in our DNA give rise to our genetic variation. When a mutation occurs in at least 1% of individuals in a population, it's termed a polymorphism. And this is a very important point because this drives a lot of our subsequent talk on clinical pharmacogenomics. The most common polymorphism is a single nucleotide polymorphism, or called a SNP. All right, And this one right here, as we'll see, is a very common one, cytosine for thymine here. Occurs when there's a difference, as the name suggests, when there's a difference in a single nucleotide. And the reason SNPs are important is because they're responsible for a large amount of our genetic variation, approximately 90%. And 2/3 of SNPs, this is interesting to me anyway, actually involve replacement of cytosine for thymine. And there's been about 10, maybe 11 million SNPs in the human genome identified so far. Certainly more to follow. And much of the characterization for clinical pharmacogenomics has focused on these SNPs, and these are the SNPs involved in drug disposition. The drug metabolizing enzymes, SNPs here, the intracellular transport of drugs, SNPs in that category as well. And I want to spend some time talking about metabolism and transport. Drug metabolism, two basic reactions, if you recall, phase 1 and phase 2. Phase 1 metabolism, this is the big cytochrome P450 system, or the CYP450 system. These 4 enzymes here, 3A, 2D6, 2C9, 2C19, these are the workhorses. These metabolized most of the drugs. Most of them are CYP3A, 2D6, and it falls off after that. Certainly there are other CYPs that metabolize medications beyond these 4, but these are the most important. These are mixed function oxidases. They produce more polar compounds. A more polar compound is going to be excreted. It's not going to be reabsorbed systemically. Phase 2 metabolism. N-acetyltransferase. These are other enzymes. Glucuronidase transferase, glutathione S transferase, and several others beyond this. These are conjugation enzymes. And phase 2 metabolism is a conjugation reaction. This increases the molecular weight and the polarity to a certain extent as well. It increases the bulkiness of these compounds. Again, a very polar, very bulky compound is not going to be reabsorbed systemically. It's going to be excreted on out of the body. Drug transporters, they're located in every organ: liver, kidney, intestines, brain, pancreas, placenta. This is a really nice review on drug transporters. So if you have any interest in this, I would consult this. Very, very complete and nice. Not necessarily a 35,000 shot view, but more like a 10 or 15,000 view. But it's excellent. There's 2 major classes. There's uptake transporters, and these are the ways that drugs and endogenous compounds get into the cells and facilitate translocation drugs into cells. And a couple of them, and as you can see, if you look at these, all these small little letters, there's more than a couple. But organic anion transport is an important part of this group. And this is a gene we'll spend some time talking about, the SILCO1B1. This is the gene that encodes that transporter. And then we have organic cation transporters as well. But there's, as you can see, there are several others up and down here in all these different tissues. Efflux, there needs to be a way for the drug or the metabolite or the endogenous compound to get out of the cell. And that's what the efflux transporters are about. These excrete drugs from within cells to the extracellular space. And some big important ones are PGP, which is paraglycoprotein, MRP2, MRP3, or others. And again, you can see several others here. So why is metabolism and transport important within clinical pharmacogenomics? The problem is, it is kind of a problem trying to figure it out. SNPs can change the protein of a CYP450 enzyme as well as a transporter, and this leads to altered drug metabolism and or transport. They can do both. And the effects on the drug disposition can lead to altered pharmacokinetics and unpredictable pharmacodynamics. So your ability to predict whether a patient has a good drug response or an adverse drug event is certainly complicated by the genetic variability within the enzymes and within the transporters as well. So now let's talk about nomenclature. You might want to grab some coffee at this point. You really have to know how to speak the language within clinical pharmacogenomics. That's why I want to spend a couple slides on this. If you're confused, see me after class, I guess. But we will try to wade through this. I'll try to make this as simple as I can. It's certainly not as bad as other systems we have in medicine. But we'll talk about the phase 1 enzymes first. And I want to start out with this simple one here. And most of the clinical pharmacogenomic action is actually on the right here. But I want to talk a little bit about this this right up front first on the left here. The CYP is considered the superfamily. The 2 is the family, the subfamily, and individual member. It's like, a microscope going further and further down. Then anything with a star and a number typically is called the allele, okay? And this actually is a suballele, so the designation can go still further. I haven't seen a suballele yet, but I'm sure that will be coming here. An allele is an alternate version of a gene. So this CYP2C9 star 2A is an alternate version of the CYP2C9 enzyme. Okay, the gene that encodes this enzyme is different. Now, typically a star 1 allele, like CYP2C9 star 1, most commonly refers to the wild type or the normal enzyme. And so far, the star 1 has been preserved. So they've kind of, fortunately, there is some consistency. I have not yet seen a star 1 yet that refers to anything other than the wild type or normal type. Star 2 or greater denote polymorphic alleles and are typically numbered in order of discovery and validation. And that's where the confusion comes in because they're just, third one discovered is number 3, 4th, 5 on up. We also further define the nomenclature by whether they're homozygous or heterozygous. The CYP2C9 star 1 star 1 has 2 copies of the wild type allele. So this is a very normal phenotype here. Heterozygous, star 1 / star 2, this is 1 copy of the wild type and actually 1 copy of a reduced function allele for the CYP2C9. So the reason we do this kind of sub-designation is because this is going to have a different phenotype than this. This is going to be an entirely normal functioning enzyme, whereas this one may be a little bit less because it has, again, 1 copy of a reduced function allele. The enzyme's not going to be as good. Phase 2 enzymes and transporters, mercifully, it's fairly similar. For UGT1A1, again, star 1 / star 1 is a homozygous for the wild type. The Silco 1B1 star 1 star 5, heterozygous, and this contains 1 functional and 1 reduced function allele. This is also called the C allele. And again, so this is where some of the nomenclature gets a little muddy because sometimes it's unclear what they're calling the C allele, which star system that is. Still other nomenclatures, they can be named by the haplotype. VCORC1, this particular SNP here, this adenine for guanine in 1639, it's called haplotype A. And if you remember, VCORC1 deals with warfarin, and I'm going to be spending some time with warfarin today. But so the SNPs can be named by the haplotype. So if we look at this system, we've got GG, which is homozygous. Again, this is the wild type normal levels of VCORC1. We've got GA, which is heterozygous, which has a little bit lower level of VCORC1. And then AA, homozygous, which has the lowest levels of VCORC1. So you'll see this designation as well. And then named by allele. Again, I alluded to it up here. The Silco 1B1 star 5 is a high myopathy risk allele. This particular transporter, especially if it's homozygous for star 5s and 15 and 16 and 17, is a very is a high risk allele for myopathy. And we'll talk about statins here in a bit. But that's what I'm referring to. So again, similar to the haplotype, TT homozygous, this is the one to be if you're on a statin, simvastatin specifically. CT heterozygous is moderate, and CC homozygous is the high myopathy risk. Okay, so we've got a bunch of little systems here, but hopefully this adds some clarity to that. Oh yeah, I always forget this one. Human leukocyte antigen, HLAs, star 5701, again. What can I say? It is a number and there's a star there, but it's certainly quite a bit different than these up here. But that's still yet another system. You'll see them all. Now, I want to spend a little bit of time on some pharmacogenomically important medications. Again, this is, as I'll stress, this is just a small grouping of them. It's more of a sneak peek at what we're going to be talking about in more detail with some of the speakers later today. But I think it gives you an idea of the breadth and scope of clinical pharmacogenomics and how common these medications are. They're ones we prescribe our patients every day, for the most part. Clopidogrel. Its Achilles heel, if it has one, is that it's a prodrug. It requires CYP2C19 metabolism to become active. There's a little picture of that little system there. And with CYP2C19, some SNPs have identified that lead to different metabolism phenotypes. We have the typical STAR1 wild type, but there's also a STAR17, which is an ultra-rapid metabolizer. And then we have the little or no function enzyme groups, the STAR2, the STAR3, and the STAR4. STAR2, STAR3 being a little bit more common than those. This is the ethnic distribution. So, and a couple references. These numbers have some variability with them, but so it's not, the point here is not an uncommon thing to have one of these loss of function alleles. 24% of Caucasians, 33% of African Americans, 50% of Asians have at least one of these, STAR2, STAR3s primarily. And this is what I also want to stress is, and why I'm talking about these medications specifically, is there have been some pretty important research that gives us a clinical signal on these. We can quibble about the research, but there is a strong signal here, and there's certainly been much more research done on clopidogrel since then. But in this JAMA article from 2009, in 227 cardiac patients, which sounds like a lot, but from a pharmacogenetic standpoint, it's actually a pretty small study. But anyway, Schuldiner and others found that 21% of clopidogrel subjects with the CYP2C19 loss of function SNPs reached the study endpoints of death, non-fatal MI, urgent revascularization, compared to only 10% without these loss of function SNPs. So there's kind of one of the early signals that we saw with clopidogrel. Warfarin, a very messy drug, but one that is still widely used today and will be continue to be widely used today. It's a messy drug, but we do know a lot about it, and we're learning more about its clinical pharmacogenomics every day. But it does consist of two isomers, an S and R warfarin. The S is the more potent. That's metabolized by this 2C9. And the R warfarin, which is less potent form, again, by a whole hodgepodge of CYPs here. The pharmacological target is VCORC1, this vitamin K reductase. We'll spend a little bit of time on that. But the important SNPs in this primarily deal with that CYP2C9, which is STAR1, which is the wild type. Again, you want to be, for most predictable action, you want to be a STAR1, but these for warfarin, and these are the extensive metabolizers. But the STAR2 and STAR3 are the poor metabolizers, and it is remarkable the drop-off in warfarin clearance you get with these. STAR1, STAR2s, or STAR3, 20 to 40% lower clearance if you're heterozygous for these. Homozygous, 50% for STAR2, and homozygous for STAR3 is 85% lower clearance. So as you could predict, these patients probably need less warfarin. And by itself, CYP2C9 accounts for about 10 to 12% of the variability. We have all these patients on warfarin, and some patients need 1 or 2 milligrams, other patients need 5 and 10 and more. And SNPs account for about 10 to 12%. And look at the ethnic breakdown here. This is in one reference of mine. Again, there is some variability as far as the distribution depending on where you look, but not uncommon in Caucasians, certainly less common in Asians, African Americans for the STAR2. STAR3 is a little rare, 7% of Caucasians and 2% in Asians and African Americans, but it is out there. The pharmacological target warfarin is also polymorphic. I mentioned this VCORC1, this vitamin K2,3 epoxide reductase. That is the inhibitory target of warfarin, and unfortunately, it also has some genetic variability there. The GG, as I mentioned earlier, is the wild type. If you're a heterozygous, you need 25% lower doses. Homozygous for the A allele, you need, or A haplotype, you need 50% lower doses. And this accounts for a lot of the variability, about 25%. And again, haplotype A, not very uncommon actually. African Americans 15%, Caucasians 42%, Asians 90%. And if you lump these two together, CYP2C9 and VCORC1 variants have been estimated to explain about 30 to 35% of the variability in warfarin dose requirements. So fairly substantial numbers here. Now I want to look at a study by Anderson, and just real quickly here, she compared two groups, a pharmacogenetically guided group, looking at and determining the genetic profile of CYP2C9 and VCORC1 in this group, fairly decent size sample size here, and a parallel control, which was a standard of care, which is you give the patient 5 milligrams or 10 milligrams and cross your fingers approach. What they found was that investigators found significantly less INRs out of the therapeutic range more time in the therapeutic range in the pharmacogenetically guided group compared to the control. So they're able to hit the warfarin targets a lot more easily than the standard of care. And they did it, more importantly, did it during this very critical 1 to 3 months of warfarin therapy when you absolutely need to have therapeutic INRs. Even more so, the pharmacogenetic guided group also had significantly lower numbers of adverse events, thromboembolism, hemorrhage, death. So again, they did a better job on efficacy, did a better job in adverse drug events. Now, the story is still out on warfarin. Feels like it's been out for years and years and years. We've got a couple, or actually more than a couple, 3 very large studies that are in various stages of ongoing and completion here that are hopefully will give us a more definitive answer on warfarin. So there's still some question there about warfarin, but the GIFT trial, the COAG trial, and the UPACT, again, will hopefully add some more precision to this specific medication. But these will be coming out hopefully within the near future here. Simvastatin. I throw this on here because I want to talk about drug transporters and the genetic variability we see with that. Drug transporters also have SNPs. As I mentioned earlier, O1B1 is a transporter for entry of simvastatin into the hepatocyte or metabolism. SOCO1B1 is that gene that encodes this specific transporter. And a genetic variant of SOCO1B1, again, this C allele, was found to be associated with an increased risk of statin-induced myopathy in a study over 12,000 subjects taking simvastatin. So this is a very nice study, very large study. And if we look at this graph over here, if you're homozygous for that CC genotype, your cumulative percentage of patients who had myopathy was certainly much greater than heterozygous. So it's amazing to me the difference that one more abnormal allele can make, but it's quite marked here. You rarely see this separation in studies for sure. Tamoxifen. I bring up tamoxifen because I need to spend a little bit of time on 2D6. 2D6 is a cytochrome P450 enzyme that metabolizes about 25% of our medications out there. So it's a very important CYP450 enzyme. Tamoxifen shares clopidogrel's weakness in that it is a prodrug. So it's metabolized by CYP2D6 to active endoxifen. And we have STAR1, STAR2, which are normal or extensive metabolizers. And I should preface this that CYP2D6 is a very complicated cytochrome. So I'm going to kind of simplify this a little bit, but there's still a lot of work ongoing with this one. But the poor metabolizers are the STAR3, 4, 5, and 6. We also have intermediate metabolizers, which are the heterozygotes, which are one normal and one poor metabolizer allele, like STAR1, STAR3. Ultra rapid metabolizers. This is called copy number variation. And in this group, there are 3 or more copies of normal alleles due to duplication. So a STAR1/STAR2/STAR2, these are the ultra rapid metabolizers. Poor metabolizers are found, again, depending on the reference, 7 to 14% of Caucasians, 14.5% African Americans. It's out there. Early stage breast cancer study in subjects taking tamoxifen found that the presence of 2 functional CYP2D6 alleles was associated with a better clinical outcome than those subjects with non-functional or reduced function CYP2D6 alleles. This is in JAMA in 2009. Again, small study, but certainly a worthwhile clinical signal to do more research on. And then abacavir, HLA-B STAR5701. There is a hypersensitivity reaction associated with abacavir. It consists of these kind of symptoms. It's not your typical anaphylaxis we think about, but it's fever, rash, constitutional symptoms, GI respiratory symptoms that worsen with continued dosing, and it can be fatal. And the symptoms will return on re-challenge, so it is a very serious one. As you would imagine, we take them off the abacavir, the symptoms get better, fortunately. And here is the breakdown, and this is the global frequency over here in this figure here. But in the US, 8% of Caucasians, 2.5% African Americans, and around 1% of Asians. Okay. And I want to stress that these are only a subset of the medications with pharmacogenomic disposition issues. I've got some statistics to throw at you. They did a survey of the top 200 prescribed medications in 2011, and they found within that top 200, 17 had pharmacogenomic information of some kind in their FDA package inserts, okay? So this tells me that there is some regulatory acceptance of clinical pharmacogenomics. And I always get a little nervous when I see drug dosing recommendations in a package insert, and yet we're still not fully engaged from a clinical practice standpoint. So that's just my take on that. But these 17 includes the 5th and the 7th most commonly prescribed medications. So these are not rare ones by any stretch. And in 2011, 362 million prescriptions were filled for these 17 medications. So the prospects or the likelihood that one patient at least, probably much more than one, of this 362 million had some type of adverse event or no efficacy from these medications based on whatever variability they had in their genotype, I think is pretty high for this, given the sheer number here. And I expect these numbers to only grow as pharmacogenomics and personalized medicine grows, definitely. So it's interesting, at least I hope it is so far, but so what? We're all here to find out how we translate all these research studies into the clinic. How do I utilize this genetic information for the patient in my clinic? That's what we're here for today. So fortunately, we have a great group, long name, but a great group, Clinical Pharmacogenomics Implementation Consortium, or CPIC. They are charged with translating genetic information into clinical actions and to make recommendations for actionable pharmacogenetic variants. They are a group of clinical pharmacologists, clinicians, and scientists, and they painstakingly review all the current literature and develop recommendations and algorithms to guide drug dosing based on pharmacogenotypes. And they re-review medications as well as new data emerges. So these things are not set in stone. So how do they pick which gene drugs to concentrate on? Well, it's based on community input, both from the CPIC group as well as the American Society of Clinical Pharmacology and Therapeutics and the public. And as I'll talk about a little bit, they're a frequent contributor to the FDA, again, the package insert issue, and they're endorsed by the American Medical Association, and the American Society of Health System Pharmacists. All right. And they looked at 14, I think it actually may be up to 16 now. They take these one at a time. There's various groups that are working on drugs different drugs simultaneously. So new drugs come out in CPT periodically. This is a list of the 14. Again, I think there may have been a couple more added to this, but a pretty good list of some very common and important medications for sure. So let's get back to our case, see if we've learned anything from this. Hopefully we have over the course of today. This is our 58-year-old female with coronary artery disease, hypertension, hypercholesterolemia. She sees her cardiologist and has a sensation of a racing heart. They do an EKG in the clinic and find that she has atrial fibrillation. And included in her treatment plan is oral anticoagulation with warfarin. She agrees to do genetic screening to look for potential variants that could affect her warfarin therapy, and this is what we get. CYP2C9*2*3 and the GAV-CORE-C1 genotype. So what do we do with those? Well, we look for the CPIC guideline for warfarin and a couple places to look for this. You can always pull it off the literature, but it's also in the package insert. And this is it. This is a recapitulation of it. So we know our patient is CYP2C9*2*3, so we're kind of in this column. And we know that she is in the GAV-CORE-C1 genotype here. So this is the starting dose that she should receive. Now, there's a lot of other factors that go into warfarin initiation dose, weight, age, etc. But we do know that the first dose she gets, if we want to avoid too high of an INR and we want to hit a therapeutic INR, shouldn't be above this range, 0.5 to 2 milligrams. So again, this is a patient that probably before clinical pharmacogenomics, we're probably would have started on a 5 milligram or 10 milligram dose, but it's remarkable how different her dose should be, and if it should be from 0.5 to 2 milligrams here. So pharmacogenomic medicine, I think it's a powerful tool. We have a lot of tools to help us pick medications for patients. renal function, hepatic function to a certain extent. Pharmacogenomic medicine, I think, is even more powerful than those because we have what it allows us to do is to pick the patient population that has a higher likelihood of having a good response. And equally important, we can pick the population and avoid those and those patients we think that might have an adverse drug event. So this is very powerful. I think it's demonstrated potential. I've alluded to and walked through a couple of the early studies. They're not the end all for those specific medications, but I think there's enough of a clinical research signal that it is time that we clinically translate this information. It has demonstrated its potential to improve efficacy and safety of medications. And again, I made this point before, and I'll make it again. As more clinical data emerges and genotyping costs continue to fall, I think there will be increasing utilization and presence in clinical medicine. All right, well, thank you.